Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing IIT JAM question paper for the subject Biotechnology conducted in the year 2023. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of IIT JAM Biotechnology, you can go to my channel Omni Bioentrance. In that, see for the playlist. In the playlist, select IIT JAM and you will find them there. First question. Which of the following compounds inhibit the polymerization of tubulin to microtubules in animal cells? ATP is not an inhibitor. Taxol. Taxol, it stabilizes the microtubule, preventing depolymerization. Thymosin, it is not involved in microtubule polymerization. Vinblastine, it inhibits polymerization by binding to tubulin. It prevents tubulin dimers from assembling into microtubules, causing microtubule depolymerization. So the answer here is option D, that is vinblastine. Then next one. Which one of the following is not a plant vascular tissue? Phloem is a vascular tissue. It transports sugars and amino acid. Periderm is a protective tissue and it is not a vascular tissue. So B is the answer. They have asked which is not a plant vascular tissue. Then stele, it is the central vascular cylinder and it contains the xylem and the phloem. So again involved in vascular tissue. Xylem is also a vascular tissue. It transports water and minerals. Then next one. A growing shoot of a germinating seedling encounters an underground obstacle. Okay. So here the growing seedling is encountering an obstacle. Which of the following hormone elicits triple response to the underground obstacle? So triple response, it is a response to mechanical stress. So here the germinating seedling is encountering a mechanical stress. So it is a triple response. And the characteristic of triple response is that inhibited stem elongation, thickened stem diameter and bending of the stem. So and the hormone that is involved in triple response is C, that is ethylene. Auxin promotes cell elongation, cytokinin regulates cell division and differentiation, and gibberellin promotes stem elongation and seed germination. So here it is triple response they have asked, it is ethylene. Then next one. Which of the following pair of antibodies contain J chain in their multimeric form? So it is IgA and IgM that contains J chain in multimeric form. Answer is option B. Then next one. Restriction enzymes that recognize the same nucleotide sequence but cleave at different position are called neoschisomers. If you see D over here, if you see in the photograph, SMA1 is GGGCCC. Similarly, SMA1, XMA1 is also GGGCCC. But SMA1 cleaves here and XMA1 cleaves here. So they have the same nucleotide sequence but they cleave at different positions. So they are called neoschisomers. What are isocodamers? Isocodamers are pair of restriction enzymes that have slightly different recognition sequence but on cleavage they generate the same termini. So MOB1 and BAMH1. Here N can be any nucleotide. GATC. BAMH1 is also having GATC but it is G here at the site of at the place of N. So it will cut here, this will also cut here, here it will cut here and MOB1 will cut here. So they are isocodamers because they have the slightly different recognition sequence but on cleavage they will generate the same identical termini. Then what are isoschisomers? They are restriction enzymes which recognize and cleave at the same recognition site. So SPH1 and BHU1, it is the same recognition sequence, CGTACG. They cleave at the same recognition site. They are isoisomers. Then next one. Which one of the following does not belong to freshwater ecosystem? Estuary. Estuary is a fresh water where forms when fresh water mixes with salt water from the ocean. So it doesn't belong to the freshwater ecosystem. Lentic is a standing water, lake, ponds ecosystem. Lentic standing water ecosystem, and lotic is flowing water ecosystem, that is rivers, streams, creeks, etc. And wetlands are marshes, swamps, logs, all these are wetlands. So the answer here is option A. Then next one. Which one of the following is transcribed by RNA polymerase 3 in eukaryote? So 3, it transcribes tRNA, 5S rRNA and snRNA. So here they have given tRNA in the option. So answer is option D. RNA polymerase 1, it transcribes 28S rRNA, 18S rRNA and 5.8S rRNA. And RNA polymerase 2, it transcribes mRNA, miRNA and snRNA. Then next one. Which of the following statements about the G1 checkpoint of eukaryotic cell division cycle is incorrect? Cell assures the existence of favorable extracellular environment. That's correct. Of G1 checkpoint. Cell assures that DNA has no damage. That is also correct for G1 checkpoint. Cell assures the damaged DNA are directed for repair mechanism. That is also correct. Cell assures complete replication of DNA. No. Complete replication of DNA occurs during the S phase, not at the G1 phase. So G1 checkpoint has nothing to do with replication. The G2 checkpoint is the one that verifies DNA replication and repair mechanism of after DNA replication. So answer here is option D. Then next one. Determine the correctness or otherwise of the following assertion and the reason. Assertion. Nitric oxide is involved in transient paracrine and autocrine signaling. Yes, that's correct. Nitric oxide is highly reactive with a lifetime of few seconds, yet can diffuse freely across membranes. That is why it is involved in transient paracrine and autocrine signaling. So both A and R are true, and R is the correct reason for A. So answer here is option A. Then next one. In mice, trait is determined by the dominant allele Y, capital Y, and the recessive allele small y. What proportion of the offsprings from a small y cross is expected to be homozygous recessive? Homozygous recessive means there should be small y, not capital Y. But here it is the F1 generation, and F1 generation you will get capital Y, small y. That is, it is not a homozygous recessive condition, it is heterozygous recessive. Heterozygous dominant, and it is dominant, not recessive. So the number of they have asked expected to be homozygous recessive will be 0. Answer is option A. Then next one. Which one of the following statement is correct about solute transport across membranes? Passive transport, they have asked the correct statement. Passive transport decrease the activation energy and does not facilitate the transport of polar compounds. Okay, that is not a correct statement, because passive transport, it facilitates movement down a concentration gradient, and it facilitates the transport of polar compounds, without decreasing the activation energy. So A is not a correct statement. Then next one. The direction in which charged solute tend to move spontaneously across a membrane does not depend on the electrical gradient across the membrane. The direction in which charged solute tend to move across a membrane depend on both the concentration and electrical gradient across the membrane. That is electrochemical gradient. So B is also not correct. All ABC transporters do not have nucleotide binding domain. Most of the ABC transporters, they have nucleotide binding domain, and it is essential for ATP hydrolysis. So C is also not correct. What is left out? P. P-type ATPases are reversibly phosphorylated as part of a transport cycle. That's correct, because P-type ATPases, they transport ions across the membrane using ATP hydrolysis, and they undergo reversible phosphorylation. Example of P-type ATPases are Na plus K plus ATPase, Ca2 plus ATPase. So D is the right answer. Then next one. Match the type of DNA repair mechanism in group 1 with the enzymes involved in group 2. So mismatch repair, MUTL, MUTS, MUTH. P4. Okay. Base excision repair, DNA glycosylase is involved. Nucleotide excision repair, UVRA, UVRB, UVRC, UVRD, Q2, R2. Then double strand break repair, RECA is involved. So the answer here is option C, that is P4, Q1, R2 and S3. Then tetracycline binds to. So tetracycline bind to the 30S subunit. So B and D cannot be the answers. And inhibits amino acyl tRNA binding. A itself is the right answer. So the answer here is option A. Then in the southern blot technique, which of the following reagent is used to detect the presence of the desired DNA fragment? Ethidium bromide. No. After southern blot technique, the membrane is treated with the probe, DNA probe, which will bind to the DNA on the membrane through complementary base pairing. So B is the answer, DNA probe. Silver nitrate is not used in staining. DNA is a DNA degrading enzyme. So answer is option B. Then next one. The pedigree given below shows individuals affected. Shaded circles or rectangles by chronic hypertension. So chronic hypertension they have specifically mentioned. So it is an autosomal disease. So C and D they have given a sex-linked dominant and sex-linked recessive. So that cannot be the answer. Assuming 100% penetrance, the inheritance of this trait is. So when you see this generation, you can see that majority of the people are having the disease. Only 2 of them are not having the disease. Okay, both the parents are affected with the disease. Now, why these two did it? So it has to do something with dominance. And I told you it is an autosomal dominant. So it is autosomal dominant is the answer. But why did you get these without disease? 2 of them without disease. That is because these 2 parents they carry this type of alleles, that is capital A small a, both of them. So the one who is carrying the homozygous recessive condition will not have the disease. So answer is autosomal dominant A. Then next one. Which of the following statements about photoproteins in plant is incorrect? Phytochromes are activated by red light. Yes, that's correct. Phytochromes are inactivated by far red light. Again correct. Cryptochromes are sensitive to blue light. Yes. Phototropins are insensitive to blue light. They are sensitive to blue light. So they have asked the incorrect statement. So D is the answer. Match the microorganism in group 1 with the human disease in group 2. Treponema pallidum. Treponema pallidum is syphilis. So P4. Okay, P4 is there only in A. So A must be the answer. Trypanosoma cruzi, it is Chagas disease. Trypanosoma gambiense is sleeping sickness. And Bordetella pertussis is whooping cough. So P4, Q3, R1, S2, A is the answer. Then next one. Which one of the following is correct in the case of conjugation of a high frequency recombination strain with F- strain of E. coli? So HFR strain has an integrated F factor in its chromosome. Now during conjugation what happens is that the chromosomal genes are transferred efficiently. But the F factor transfer occurs only when the integrated F factor is excised from the chromosome. And this situation of the F factor getting excised from the chromosome is very, very rare. So what result do you get? You get recombinant F- cells with the new chromosomal traits. But F- plus cells will be very, 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 very low, or it will not be there at all, because F factor transfer frequency is very low. So recombination frequency is high. Yes, that's correct. A is correct. F factor transfer frequency is low. A is the answer. Recombination frequency is high. That's correct. F factor transfer frequency is also high. That is not correct. Recombination frequency is low. Recombination C and D are not correct. Then next one. Determine the correctness or otherwise of the following assertion and the reason. Assertion. Cardiovascular organization called double circulation provides vigorous flow of blood to the brain, muscles and other organs. Yes, that's correct. The blood is pumped a second time after it loses pressure in the capillary beds of the lungs or skin. That is also correct. And it explains the assertion. So both A and R are true, and R is the correct reason of A. So that's it for now. Thank you for watching my channel. Please do like, share and subscribe if you are new to my channel. If you want still previous year question papers of IIT Biotechnology, you can go to my channel Omni Bio Entrance. In the playlist, select IIT JAM and you will find them there. Thank you.